is protective against diet-induced obesity and attenuates adipocyte hypertrophy and inflammation at different fat depots. Obesity is defined by the World Health Organization as an excessive accumulation of fat that has the potential to impede on health. One is said to be obese when the BMI or body mass index is equal to or greater than 30. In 2016, approximately 13% of adults worldwide was said to be obese. Obesity has a multifactorial etiology, which could be a result of lifestyle factors such as eating an unhealthy diet or maintaining a sedentary lifestyle with minimal to no physical activity. It can also be due to genetic factors such as mutations in the melanocortin 4 receptor gene or in the leptin receptor gene. Obesity is associated with the development of cardiovascular disease, non-alcoholic fatty liver disease, type 2 diabetes, osteoarthritis, and cancer, to name a few. The reason for this current study is due to previous findings in a laboratory by Dr. Miller and team who were trying to understand the process of adipogenesis and identifying molecular factors which could be important in regulating this process. What they did then was to collect white adipose tissue from adult obese donors. They then isolated adipose-derived stromal cells from this tissue. Cells were induced to undergo adipogenesis, and RNA was isolated from these cells to perform a transcriptome analysis. They identified several novel genes, including the SLC7A8 gene, which had not been previously described in the context of adipogenesis or obesity. The SLC7A8 gene is a solute carrier family 7-member H gene located on the long arm of chromosome 14. It is a transmembrane transporter of neutral amino acids, and mutant form of this gene has been associated with age-related hearing loss, as well as aminoaciduria, which is an accumulation of amino acids in the urine. To date, the role of this gene in adipogenesis or obesity development is unknown. The aim of this study is therefore to elucidate the role of the SLC7A8 gene in diet-induced obesity. To begin the study, wild-type and knockout mice were equally divided and fed with either a control diet or a high-fat diet for a number of weeks. At 5 and 14 weeks of experimental feeding, the mice were terminated and tissue was isolated for further analysis. The objectives of the study were to assess the effect of glucose and insulin on metabolism, to measure weekly body weight, to perform glucose tolerance tests, to conduct histological analysis, and also to perform immunohistochemical analysis. Before introducing mice on their experimental diet, glucose tolerance test was performed. This is normally done to measure how the body responds to glucose, and in an obesity phenotype, this can be used to predict a pre-diabetic state. Glucose levels were measured at time 0, and thereafter exogenous glucose was administered, and the glucose levels were monitored for a period of 120 minutes. The glucose levels increased from time 0 to 30 minutes and started declining thereafter. At the end of 120 minutes, the glucose levels for both the wild-type and knockout mice were similar. This indicates that the metabolism of glucose by these mice is similar irrespective of their genotype. Following GTT, insulin sensitivity test was performed. Insulin functions in regulating blood glucose levels, and therefore an IST would then be conducted to measure how sensitive the body cells are to insulin. Similar to GTT, glucose levels were measured at time 0 and exogenous insulin was administered, and glucose levels were monitored over a period of 120 minutes. At the end of this time frame, it is observed that the wild-type and the knockout mice have similar glucose levels, indicating that insulin metabolism is similar irrespective of the genotypes of these mice. The mice were introduced to the experimental diet, and body weight was measured over a 14-week period. The results on the graph indicate that the wild type on high-fat diet gained significantly more weight than the wild type on control diet. The knockout mice on a high-fat diet also gained significantly more weight than the knockout on the control diet. When wild type on high-fat diet were compared to the knockout mice on the high-fat diet, it can be seen that the wild type on high-fat diet gained significantly more weight, which was visible from week 3. No differences in weight gain were seen between the wild type on control diet and the knockout on control diet. Glucose tolerance tests were performed 5 weeks and 14 weeks post-experimental feeding. The 5 weeks results indicated by the graph on the left indicates that no significant differences were observed in the metabolism of glucose across the 4 categories of mice that were under study. After 14 weeks, it is shown that the wild type on the high-fat diet had significantly higher levels of glucose in their blood when compared to their wild type on control diet counterparts. But also when you compare the wild type on high-fat diet with the knockout on high-fat diet, you can see that the glucose levels were higher, which were observed from 30 minutes across the 120 minutes. At 120 minutes, the glucose levels of the wild type on high-fat diet were still high, indicating that the metabolism of glucose in these mice were quite slower, which is a sign of glucose intolerance in these mice. When you look at the knockout on high-fat diet, you can see that at the end of 120 minutes, the levels of glucose have gone almost back to those at baseline, indicating that these mice are able to metabolize their glucose and do not show a glucose intolerance in terms of their metabolism. Following 14 weeks of experimental feeding, mice were terminated and their organs were collected and weighed. The results on the graph indicate that the wild type on high-fat diet mice had significantly larger subcutaneous adipose tissue, perigonadal or abdominal adipose tissue. They had larger mesenteric adipose tissue, which is the adipose that lines the intestines. They had significantly larger interscapular brown adipose tissue and significantly larger liver when compared to the wild type on control diet and the knockout on the high-fat diet. These results of the weight correlate to the large body weight seen in the wild type on high-fat diet. Histological analysis were performed on perigonadal adipose tissue, and the results indicate that the wild type on the high-fat diet had significantly larger adipocytes when compared to the knockout on high-fat diet and the wild type on the control diet. Histology of the subcutaneous adipose tissue also indicates that the wild type on the high-fat diet had significantly larger adipocytes when compared to the knockout on the high-fat diet and the wild type on the control diet. Histology on the mesenteric adipose tissue indicates that the wild type on high-fat diet also has significantly larger adipocytes when compared to the knockout on the high-fat diet and the wild type on the control diet. Previous research has indicated that when white adipose tissue expands and eventually reaches its capacity to store lipids, these lipids will be redirected to neighboring peripheral organs, such as the liver or the muscles. This can then lead to a range of metabolic syndromes or other health complications. The histological analysis of the brown adipose tissue indicates that the wild type mice on the high-fat diet have visibly larger lipid droplets when compared to the knockout on high-fat diet and the wild type on the control diet. The brown adipose tissue contains a lot of mitochondria, and it is important for the production of heat and fatty acid oxidation. it has been shown that in obesity, there is mitochondrial dysfunction, which is due to a production of excessive reactive oxygen species, which can then cause oxidative stress. The liver histology indicates that the wild type on high fat diet has lipid droplet accumulation, which is not observed on the knockout on high fat diet and the wild type on the control diet. When one looks at the wild type on high fat diet, there are two types of lipid droplet accumulations which can be seen. One is macrovesicular steatosis, indicated by the arrow, which is just big lipid droplets across the tissue. And the second type is the microvesicular steatosis, indicated by a circle, which is the accumulation of lipid droplets in the cytoplasm of the hepatocytes. Both these types of lipid droplet accumulation have been shown to be the early signs of the development of non-alcoholic fatty liver disease. The bottom panel shows the central veins in the liver. The wild type on high fat diet shows a lot of blood cells in this vein, which is known as hepatic congestion. And this is due to an impairment of the drainage of the liver. And this is not seen in the knockout on high fat diet and in the wild type on the control diet. Following histology, immunohistochemistry was conducted to determine macrophage infiltration into these tissues. The F480 marker was used to analyze these macrophages and to explain the state of inflammation in these tissues. When the wild type on high fat diet was compared to the knockout on high fat diet, the perigonadal adipose tissue showed that the wild type on high fat diet had significantly higher macrophages when compared to the knockout on high fat diet, indicating that there was higher inflammation in the wild type on high fat diet mice. In the mesenteric depot, the wild type on high fat diet showed again significantly higher levels of macrophages when compared to the knockout on high fat diet. The staining for the macrophages is indicated by the brown color in these images. And when the subcutaneous adipose depots were compared, the wild type on the high fat diet showed to have significantly higher macrophages when compared to the knockout on high fat diet. Looking at the liver shows higher staining of the wild type on high fat diet when compared to the knockout on high fat diet. Again, also indicating that there's higher inflammation on the wild type on high fat diet mice. In conclusion, the results presented indicate that deleting the SLC7A8 gene does not have any effect in glucose and insulin metabolism when mice are fed on a normal diet. Deleting SLC7A gene shows that the mice are protected against diet-induced obesity and glucose metabolism is improved. SLC7A8 gene deletion attenuates lipid accumulation in different adipose tissues and also reduces the inflammatory profile in these tissues. Deleting this gene also shows that liver steatosis is attenuated, which could indicate that these mice could be less susceptible to developing metabolic syndrome. I would like to thank my supervisors, Dr. Melvin Ambele and Professor Michael Pepper, who have been supportive throughout the progression of this project. Mr. Musha Vengwa and Ms. Ilse van Rensburg, who assisted us with all of the animal work. To Mrs. Marlene van Heerden, who assisted with all the histology and immunohistochemistry. And finally, a very huge thank you to our funders, the National Research Foundation, the National Health Laboratory Service, University of Pretoria, and the South African Medical Research Council for all the funding that they provided and making this project a possibility.